Hi, my name is Danielle Winchester, and I am the NIH Commons Fund's Communication and Evaluation Liaison for the Kids First program. The last part of the webinar will focus on frequently asked questions we have received, including a brief overview of the Kids First and the different components of the program. One of the first questions we get, how did Kids First get started? The program was initiated in response to the 2014 Gabriella Miller Kids First Research Act. The act was signed into law just 5 years ago. The bill transferred $126 million that was in a presidential nominating convention fund to a Pediatric Research Initiative Fund and authorized the appropriation of $12.6 million per year to the NIH Commons Fund for 10 years. The first appropriation was in 2015. Gabriella Miller, which you have heard, the bill and the program's namesake, was a 10-year-old tenacious advocate for pediatric research who have lost her battle with DIPG just 6 months before the bill was signed. The vision of the program is to alleviate suffering from childhood cancer and structural birth defects by fostering collaborative research to uncover the etiology of these diseases and supporting data sharing within the pediatric research community. Collaborative research and data sharing are two key values that we feel will lead the way to achieving the ultimate goals of improving diagnosis and treatment for patients with these conditions. We are often asked, why is it important to study childhood cancer and structural birth defects together? The strength of studying these pediatric conditions together is supported by the fact that a risk factor for childhood cancer is being born with a birth defect, suggesting there are shared genetic pathways underlying these conditions. This graph shows a combination of specific cancers that are associated with specific birth defects. However, both sets of conditions tend to be relatively rare and therefore warrant coordinated efforts to combine, build, and study sufficiently powered data sets. This slide lays out who is involved in the Kids First program. Kids First is a NIH effort supported by the Commons Fund. Each Commons Fund program is overseen by an NIH working group. The Kids First working group is chaired by institute leaders from NICHD, NHLBI, NCI, and NHGRI, with representation from 11 other NIH institutes and centers, as well as the CDC. The Kids First program has implemented 2 major initiatives thus far. You have heard quite a bit about the DRC. Before the DRC was launched, we started the program with a large-scale data generation effort to identify childhood cancer and structural birth defects cohorts for whole genome sequencing by the Kids First funded sequencing centers. And in the next few slides, we will tell you more about each initiative. We have discussed and showed you features of the portal, but I would like to add that the Data Coordination Center and the Administration and Outreach Core are important components of the DRC. I mentioned previously, for our first initiative, we funded sequencing centers to perform the DNA sequencing of pediatric cohorts. For the last 3 years, we have worked with Hudson Alpha in collaboration with St. Jude and the Broad Institute. And in the first year, we worked with Baylor and WashU. Together, these sequencing centers have or are currently sequencing a total of 26,000 genomes. Now I will turn it over to Valerie, who will share with you some information about the cohort.
You probably noticed that the question, who is part of Kids First, is taking many slides to explain. So I'll tell you a little bit more about our EXO-1 cohort project. So in the first 4 years of the program, we have selected about 26 projects or cohorts representing approximately 26,000 genomes, as Danielle described. That also represents an estimate of about 10,000 cases or patients for sequencing, and these are all at various stages of the pipeline. 7 of these data sets from these projects have already been released, which means that the data are available for analysis after approval, as you saw from Allison and Vincent's presentation. Each XO1 project is led by a team of investigators who provide samples from a cohort for sequencing, data sharing, and analysis. The cohorts or projects are roughly organized by condition, as you see here, but some projects study multiple conditions. For example, one project is made up of children who were born with a birth defect and then developed a childhood cancer later in life. So this project is made up of a variety of birth defects and childhood cancers. Some cohorts are also sequenced across multiple years, and this is because, based on our limited funds, we are often not able to sequence one large cohort in one given year. NIH program staff are thrilled with the diversity of conditions that continue to come in through our XO1 sequencing opportunities. It's enabling us to build a diverse and strong pediatric data set, and it also draws in a variety of expertise from these research teams. Last year, in fiscal year 2018, we were able to expand our sequencing efforts with support from other NIH programs. With the help from the Trans-NIH INCLUDE project, and INCLUDE stands for the investigation of co-occurring conditions across the lifespan to understand Down syndrome, we are collaboratively sequencing a cohort studying children with Down syndrome and congenital heart disease, or acute lymphoblastic leukemia, and another cohort of children with Down syndrome and myeloid malignancies. In collaboration with NIAAA, we are sequencing a cohort that aims to uncover the role of genetic susceptibility to fetal alcohol spectrum disorders. We truly feel that by working together, we can more rapidly foster genetic discovery, and we are really excited to actually demonstrate that with these projects. We have also recently established a collaboration with the Knockout Mouse Phenotyping Project, or KOMP2, which is another Common Fund program. Although, as the name suggests, KOMP typically produces knockout gene models, this collaboration is focused on KOMP sites developing mouse strains to study, phenotype, and validate coding and non-coding genetic variants identified from Kids First datasets. So far, investigators have proposed variants from these 4 highlighted datasets, and you can learn more about these projects and the variant nomination process at the link that's on this slide. We are continuing to accept proposals on a rolling basis for investigators to nominate variants based on Kids First data until May 31st or until the funding for this pilot project are committed to projects. That said, we strongly encourage any researchers to collaborate with KOMP to pursue collaborations even outside of this pilot. As you've seen, we are always looking for ways we can work together with other efforts in the spirit of accelerating pediatric research. But we also want to point out that within the program, the XO1 cohort investigators, sequencing centers, and the Data Resource Center also collaborate with each other, in some cases to address specific scientific questions or analyses within or across projects. And the impact of the program is amplified by the collaborations that each investigative team has already established. For example, several Kids First XO1 investigators are part of international collaborations to study specific pediatric conditions. Kids First sequencing centers are often involved with other large-scale data efforts to inform other Kids First projects, and the Data Resource Center is charged with facilitating new collaborations as well as engaging with patients, families, and foundations. So each component of the program is designed to draw in more researchers and expertise as we continue to build a Kids First research community. But of course, we also consider any researcher who makes use of or makes discoveries with Kids First data to be part of this community, which leads to our next frequently asked question, which is, how do I access data? As you saw from today's demo, anyone can register and log into the portal to search, filter, or visualize information about Kids First datasets. But in order to access individual level sequence data, investigators must submit a data access request through dbGaP, which will be processed by our Kids First Data Access Committee. After approval, data sets can be accessed and analyzed through the cloud-based workspaces that are integrated with the Kids First Data Resource Portal. Last September, we hosted a day-long webinar with the Data Resource Center, and one of these presentations went over lots of detail about how to submit a data access request. So we actually encourage you to watch that separately, and there are also other webinars from that day that may be of interest to you. And now I will turn it over to James Coulomb.
So a question that we've been asked is how does Kids First support data sharing? And first, I want to stress that as part of stimulating and accelerating pediatric research, we consider data sharing to be extremely important, and we've taken several steps to support data sharing. First, when selecting cohorts for sequencing, a priority for us are cohorts whose data can be shared and analyzed in combination with other data from other conditions, and because all data sets are not consented that way. We created and have posted FAQs to explain the genomic data sharing process and clarify our expectations for data sharing in the Kids First program.
Kids First frequently asked questions are available at commonfund.nih.gov/kidsfirst.
To speed up access to Kids First data, we formed a separate Kids First Data Access Committee to streamline these data access requests. We are also discussing with other NIH programs ways to facilitate data access across programs. And of course, as you have seen, we've funded the Data Resource Center to build an internet portal and some very impressive tools to facilitate data analysis and analysis across data sets. Oops, what did I do? Push that. There. Okay. Another question that comes up is how are Kids First data sets selected? First, researchers apply to have their cohort samples sequenced by the Kids First sequencing centers through the EXO-1 mechanism. These EXO-1 proposals are discussed and scored by a scientific peer review panel, and then the Kids First working group makes selections based on a number of criteria. We consider the scientific and technical merit based on the peer review scores and written critiques from the review panel. We consider broad data sharing and ability to combine data across conditions, as was mentioned before, is a very important factor in our selection process. And we try to evaluate the perceived overall data, value of that data set for the Kids First data resource, and try to balance our investments in childhood cancer and birth defects. We also consider the availability of the samples and the quality of the samples and their suitability for sequencing. Okay, a frequent question is what funding opportunities are available to support Kids First research? You can apply to have samples sequenced through the EXO-1 process, and the funding opportunity announcement shown here is out of date, but we expect to reissue this announcement for the next 2 years, although this announcement is a good guide for preparing those applications so you can look ahead to see what we're asking for. You can also apply through the Kids First R03 announcement for analysis support or through other mechanisms such as the NIH parent R01. In order to find out about future Kids First funding opportunities, I strongly suggest that you sign up for the Kids First listserv at the address shown here.
Join the Kids First listserv at commonfund.nih.gov/kidsfirst/register.
Yep. James discussed how funding opportunities that are available for the Kids First program, but for community members, healthcare professionals, patients, and family members, if you would like to get involved with the Kids First program, you can search data that's available through the Kids First Data Resource Portal, which is at portal.kidsfirstdrc.org. You can also connect with and provide feedback to the DRC by sending an email to support@kidsfirstdrc.org. You can contact the program for questions directly or feedback by sending an email to kidsfirst@od.nih.gov. For additional information about the Kids First project, you can visit the Kids First EXO-1 project page and read each abstract. If you want to learn more about the Kids First investigators, we encourage you to view their profiles on the Kids First DRC website. For more information about available data sets that Valerie discussed, you can visit the Kids First DRC support page. And lastly, these are helpful links for you to visit after our presentation. The first one is the Kids First program website, which is commonfund.nih.gov/kidsfirst. The second is the Kids First DRC website, which is kidsfirstdrc.org/. And lastly, you have the portal website, which is portal.kidsfirstdrc.org.